Welcome to The Explainer. Today, we're tearing right into the dense, honestly fascinating microbiology of coronaviruses. But, think of this less like a dry biology lesson and way more like the ultimate microscopic heist movie. We're talking master burglars, completely bypassed alarm systems, and a totally wild biological arms race. All right, let's just dive right in. So, the core mystery we're trying to solve today is pretty profound. I mean, we've lived with human coronaviruses for literally eons. We're talking about those strains with totally forgettable names like 2290 or OC43. You know the ones. They circulate, give you the common cold, maybe you get the sniffles for a few days, and life goes on. But then, along comes the novel SARS-CoV-2. How exactly did a family of viruses famous for giving us a mild, scratchy throat suddenly get the tools to shut down the entire globe? To really wrap our heads around this threat, think about the last time you had a mild cold. Evolutionarily speaking, a good pathogen doesn't actually want to kill its host, right? Traditional strains have adapted to us over centuries. They stick to our upper airway, pull up some mucus to make us cough, and spread just fine without doing catastrophic damage. But SARS-CoV-2, this was a completely novel strain that jumped species. It doesn't just hang out in the upper airway. Instead, it aggressively hunts for a specific receptor called ACE2, and that receptor is found everywhere, not just in our noses, but buried deep within our lungs and our blood vessels. And honestly, size really does matter here. When you look at the coronavirus genome compared to other common respiratory bugs, it is just massively overprepared. Influenza and RSV, they make do with just 11 proteins, but human coronaviruses pack a whopping 26. To put that in perspective, if the flu shows up to a break-in with a simple crowbar, this virus brings a massive, highly specialized duffel bag full of lockpicks and tools designed to replicate and completely suppress your immune system.
Section 1. The perfect break-in. Bypassing the cellular locks. Let's see exactly how it gets inside. The way this virus actually gets in is remarkably deceptive. First up, the outer section of the virus's spike protein, we call that S1, attaches to our cell's ACE2 receptor. Basically, it grabs the cellular doorknob. But turning that knob, that requires a pretty sneaky trick. The virus has this short amino acid bridge connecting its S1 and S2 sections, and this bridge is perfectly designed to be cut by a naturally occurring human enzyme called furin. It's literally using our own enzymes as a lockpick. Once that bridge is cut, the S2 section activates, the virus fuses its membrane with ours, and bam, it slips right inside the cell. Moving on to Section 2, the microscopic battlefield, alarms and evasion. So once the virus breaks in, its positive sense RNA gets right to work at our ribosomes, immediately copying itself. But here's the thing, our cells are not defenseless bank vaults. They come equipped with these incredible redundant chemical sensors. See, humans don't naturally create double-stranded RNA, but viruses absolutely do. So when the virus replicates, sensors like MDA5 immediately clock that foreign RNA. At the same time, RIG1 is detecting these unique chemical caps, and we've even got toll-like receptors acting as redundant backups. The split second these sentinels detect a breach, they trigger a massive chemical cascade. And when those alarms trip, they trigger the release of something called interferons. This is the innate immune system's absolute most potent early-stage weapon. What's fascinating about interferons is that they don't actually kill the virus directly like an antibiotic would. Instead, the infected cell shoots these chemical messengers out to all the neighboring uninfected cells. It's exactly like pulling a biological fire alarm. It tells all the neighbors, hey, gear up, shut down your factories, and start manufacturing substances that will destroy this viral RNA before it can spread to you. Now, pretty much all our nucleated cells can produce interferon, sure, but there is one absolute heavyweight champion in this fight, the plasmacytoid dendritic cell, or PDC for short. These highly specialized cells are the grand champions of our early defense. Why? Because they can crank out up to 1,000 times more interferon than a normal cell. They are literally the ultimate bridge between our quick innate response and our long-term immunity. But hey, remember that massive viral toolkit we talked about earlier? Well, the virus uses some incredibly sophisticated camouflage to fight back. As soon as it starts replicating, it essentially hijacks the host into building a double membrane vesicle right around the viral RNA. It basically pitches a highly secure tent inside your own cell. By hiding inside this vesicle, it completely conceals its genetic material from our RIG1 and MDA5 sensors, which drastically slows down those interferon fire alarms. And the crazy part? The virus doesn't just hide. It actively sabotages our defenses. It dedicates a huge chunk of its genetic arsenal to shutting down the chemical pathways that tell the nucleus to even make interferon in the first place.
And if that wasn't enough, it drastically reduces the number of circulating natural killer, or NK, lymphocytes. Usually, these NK cells with spot an infected cell inject it with enzymes and just destroy both the cell and the virus inside. So by liquidating our NK cells and scrambling our chemical messengers, the virus buys itself the precious time it needs to multiply. Which brings us to Section 3, the collateral damage, why some get sicker.
So, if the innate immune system can't clear the virus out of the upper airway during those first few critical days, the situation gets really dangerous. Older individuals naturally just have fewer of those highly active PDCs we talked about, as well as reduced natural killer cells. On top of that, people with comorbidities that cause chronic inflammation actually have immune systems that are deliberately dialed down to prevent flare-ups. Because of these weakened defenses, the virus survives that initial standoff, packs up, and relocates deep into the lungs, where its absolute favorite target, that ACE2 receptor, is just incredibly abundant.
This frontal attack in the lungs is where the true devastation really sets in. The immune system basically panics and turns against itself, triggering a dangerous, totally out-of-control inflammatory response that you've probably heard of, a cytokine storm. Immune cells, particularly neutrophils, which ironically aren't even that helpful against viruses, just flood the area. This massive inflammatory panic ends up destroying the delicate endothelial lining of the blood vessels and severely damages the lung tissue. The immune system is essentially nuking the very battlefield it's trying to save. Section 4. Building the ultimate weapon, the engineered blueprint. Faced with all of this, traditional vaccine methods just simply weren't going to cut it. I mean, old-school vaccines rely on growing massive amounts of a virus in a lab and then either killing it or weakening it through hundreds of tissue passages. That process literally takes years. Plus, SARS-CoV-2 doesn't even grow very well in laboratory tissue cultures anyway. This extreme urgency forced a historic leap to innovative mRNA technology. We basically had to construct a synthetic defense completely from scratch. The absolute brilliance of the mRNA solution really boils down to two main components. First, scientists engineered a synthetic single-stranded mRNA that carries the exact genetic code for exactly 1,273 amino acids. That is the precise recipe for the viral spike protein. But if you inject bare RNA into the body, it gets destroyed instantly. So second, they wrapped it in these tiny lipid nanoparticles. Think of it like a multi-layered smuggler's coat made out of phospholipids and cholesterol. It protects the RNA from our own enzymes and helps sneak it right into our cells, delivering the blueprint straight to our ribosomes. And here is why that is a total game changer. Because we're only delivering the recipe for the spike protein, we completely skip all those dangerous viral evasion proteins. The cell manufactures just the spike, and our immune system immediately spots it as foreign material. We are safely teaching the immune system to recognize and completely neutralize the burglar's master key, generating these potent antibodies long before the actual dangerous virus ever even shows up at the front door.
But, as absolutely incredible as our scientific response has been, this story is far from over. SARS-CoV-2 is now completely endemic. While most of the world has some level of immunity, this virus has a notorious mutation rate. Because of its uncanny ability to mutate and recombine its genetic material, constantly tweaking that spike protein to find new ways past our defenses, we really have to ask ourselves, what is the next chapter of this biological heist going to look like? And more importantly, how will our engineered blueprints adapt to stay one step ahead? Definitely something to chew on until our next explainer.